Thank you so much for inviting me to give this talk today. And thank you so much for holding this as a hybrid meeting. My kids started school today, so there was no way I could have come to Oxford. I actually still have a lab at the University of Copenhagen. So I'm juggling two research groups, one in the US and one in Denmark. So I'll be talking to you about venom-inspired design of novel insulin therapeutics. But before I start, I want to acknowledge everybody involved in this work, so I don't forget in the end, people from my group and also from Toto Oliveira's lab at the University of Utah, other collaborators in the US, particularly Denny Cho, friends and collaborators in Australia, Germany, and the Philippines. And so what my lab is mostly focused on is these toxins that we call doppelganger toxins. And doppelganger toxins are basically toxins that mimic endogenous signaling peptides of the prey or predators. And those signaling peptides could be hormones or neuropeptides. The term doppelganger toxins we chose because of its German origin. It basically means a lookalike. So it's something that looks like something else. And then I guess in this particular case, it's hard to say who the original toxin was. The lab, our group mostly works on cone snail venoms, but we have recently also started to look at other venomous animals and also at viruses or pathogens and parasites, because those organisms also make toxins to manipulate the physiology and behavior of the prey or host. And those are also sometimes doppelganger toxins. And the particular doppelganger toxin I want to talk to you about today is this insulin we found in Conus geographus that mimics the endogenous insulin in fish prey. And because insulin in fish is very similar to insulin in human, we were able to explore this compound for drug design for diabetes. So I realized that maybe not all of you are familiar with cone snails. So let me introduce Conus geographus to you. Conus geographus is an unusual cone snail species. It's one of the two known cone snails that releases venom into the water. So you can see how it opens this large fake mouth. It releases venom into the water, and in this particular case, it immobilizes a large fish. But we have seen Conus geographus do the same with a whole school of small fish. So by releasing venom into the water, it catches its prey. And this is a complex mixture of different components. And we found a few years back that insulin insulin toxin was part of this mixture. So once the prey is captured, Conus geographus can inject venom through a harpoon like a radula tooth, and then it walks away or crawls away and digests its prey. And before I can tell you about the very unusual features of the venom insulin, I need to tell you a little bit about insulin itself. And most of you will be familiar with this since insulin is one of the most important human hormones. So it's a peptide hormone secreted by pancreatic beta cells. It's of critical importance for glucose homeostasis. So it induces glucose uptake from the blood into several tissues. So without insulin production or insulin injection, we would starve because we would not be able to utilize the glucose that we get with our food or drinks. Structurally, insulin is a two-peptide chain hormone. So there's an A peptide chain and a B chain connected by two disulfide bonds. And the A chain has an additional intramolecular disulfide. And insulin in humans and fish is very similar. So you can extract insulin from fish and treat human disease. And insulin impairment, so the inability to produce or respond to insulin, is characterized by diabetes. There are various types of diabetes, and the two major types are type 1, which is an autoimmune disease, and type 2, which is of obesity or lifestyle-related diabetes. And insulin therapy, which is daily insulin injections, aims at restoring normal glucose levels in people with diabetes. And it's the only available treatment for people with type 1 diabetes, for many with type 2, and some with pregnancy-related diabetes. And so in the insulin effect in your body is that if you have breakfast in the morning, insulin secretion goes up and then comes down to a baseline level. If you sit in front of your computer now and you're having a snack or a drink, insulin will go up again and go down again and so on. And the way it's released from the pancreas, and this is important for the purpose of my talk, is that it's released as a hexameric compound. So it's 6 insulin molecules that dissociate into 3 dimers and then the monomer. And it's only the monomer that is active at the insulin receptor and then induces glucose uptake. And this can be a rate limiting step. So hexamer to dimer to monomer conversion can be slow. And so if you have diabetes, it's actually not easy to reconstitute this insulin effect. So if you don't produce your own insulin, how do we make sure that glucose levels are normal and insulin is released in or injected in the same way? And so the pharma industry has designed these long-acting insulin analogs that give you a baseline level throughout the day. And then in addition to that, ideally there would be these rapid-acting insulin analogs that you can take whenever you have a meal. And these long-acting insulin analogs have been very successfully made, and it's those rapid-acting analogs that I'll be talking to you about, because as of today, there's no truly monomeric rapid-acting analog. So ideally, something really fast-acting would be monomeric. So it wouldn't have to dissociate from the hexamer into the dimer and the monomer. It should already be monomeric. And the reason for why no monomeric compound is available yet, despite decades of research, mostly in pharma, is shown on this slide. So here you see the sequence again of insulin, and it's this region of the bead chain that is highlighted here and also in the structure of a single insulin molecule that very quickly dimerizes with another insulin molecule. So this region of the bead chain makes sure that there's a dimer, and then 3 of those dimers form the hexamer. So most of you will probably now think, well, let's just remove this region of the bead chain. And if we remove it, then we should have a monomer because we destroyed that dimer interface, and that is true. But when you do that with human insulin, you almost lose all of its activity at the human insulin receptor. So here you see an activation curve. This is the full active native human insulin. And if you make the analog that gets to the receptor very quickly, it can no longer properly activate the insulin receptor. And this has been known for decades that you cannot remove that region of the insulin bead chain. And structurally, it has also been solved. It is known now that this region interacts with the insulin receptor in critical ways. And so when we found the venom insulin, which we call insulin G1, this is the compound we found. This is the X-ray structure. This is the sequence. There's an A chain and a B chain. Structurally, it looked very, very similar to human insulin, but what you may see fairly quickly is that that region that is involved in dimerization is missing. And so we knew at that point, and we experimentally determined this later, that this compound was truly monomeric, so it was a fast-acting insulin. But would it be active at the human insulin receptor? So when we tested it, to our great delight, we could see that although it wasn't as active as human insulin, it was active enough to show that indeed one can make, or in this case, nature can evolve monomeric insulin compounds with potent activity at the human insulin receptor. And so at this point, we knew that Conus geographus, this fish hunting cone snail, had evolved a monomeric fast-acting insulin to induce hypoglycemic shock or insulin shock in its prey. And this is a well-described phenomenon in humans. If you accidentally give yourself an insulin overdose, in mild cases, that this causes dizziness. In severe cases, it can cause coma and death. And of course, we had to show this in animal models of the disease. So we tested several models of diabetes in mice and also in fish. And we could see that the venom insulin fairly potently and quickly lowers blood glucose levels. And because this is a net hunting cone snail that releases venom, we also wanted to show that if you take a zebrafish larvae and you release the venom insulin into the water, the fish could no longer properly swim. Every time it tried to swim, it would have these seizure-like movements, whereas a fish without a venom insulin exposure was happily swimming away. Okay, so we made this discovery of this monomeric fast-acting insulin, and we very briefly discussed whether we should develop this as a new drug lead for the treatment of diabetes. And this was work done with Toto Oliveira and Danny Cho, who was at the University of Utah at the time. And Danny's one of the very few people in academia who works on insulin analog design. And so we kind of dismissed that initial idea, but this is the idea we pursued instead. So our idea was, could we take human insulin, make it monomeric, so remove that region. So you may remember that you get a monomeric compound, but this compound is no longer active. But then could we take amino acids from the venom insulin, put them onto human insulin and make it active again, thereby creating a venom-inspired analog of human insulin that would now be truly monomeric, which, to remind you, is something that nobody had achieved at that point. And so in order to do that, we teamed up with Mike Lawrence in Australia. And Mike at the time had this construct of the so-called microinsulin receptor. So it's a region of the insulin receptor shown here that harbors the primary binding site of insulin. And with that microreceptor construct, we could determine the X-ray structure of this complex between the venom insulin and the microreceptor. So the insulin receptor shown in gray, the venom insulin is shown in blue, and human insulin is shown in red. And what we were interested in was this region here of the B chain that was known to be important for receptor activation, and what amino acids were present in the venom insulin that would now come in and basically serve as surrogates of this missing region. And we identified a few amino acids. And most importantly, we basically identified the minimum binding motif. So how small an insulin can be to still be able to activate the human insulin receptor. And one of the big names in insulin research, Pierre de Maitz, said in a News and Views article that 95 years after its discovery, the insulin molecule still has many secrets to reveal. And I show this because insulin is probably the best studied human hormone, but if you find a doppelganger toxin that mimics an endogenous signaling peptide, in this case insulin, there's always new things you can discover because the toxin is under very different selection pressures than the endogenous hormone. And so with that information, we basically went back to the idea that we had. We knew which residues, which amino acids were important in the venom insulin. We took human insulin, made it mono. Then we put those residues on that human insulin, thereby creating a monomeric human insulin. And when we tested it at the insulin receptor, you can see this line shown in red here, it had full activity. So we really were able to create a truly monomeric insulin analog that was inspired by this venom insulin. And this has been licensed and is currently in preclinical studies. So we're hoping it'll one day make it into the clinic as the first monomeric insulin. And as many of you know, there are thousands of barriers a compound has to overcome before it can reach the clinic. So even if our compound never makes it into the clinic, where we know that we have inspired other research labs and also pharma to look into revisiting how to make a monomeric insulin analog. So this is a review written on the future of type 1 diabetes treatment last year, and it highlights the discovery of the venom insulin because it exists in a monomeric form, which is a key characteristic for the rapid absorption from subcutaneous space. And so very quickly after we found the venom insulin in Conus geographus, of course, we wanted to know, do other cone snails also have venom insulins? And this was done in an initial study where we just looked at 16 cone snail species, including fish hunters, snail hunters, and worm hunters. And only fish hunters make insulins that look like the fish hormone, and snail and worm hunters make insulins that look like the invertebrate hormone. So we knew that we had to sequence many cone snail species, including many fish hunters, to find more of these unusual compounds. And so today we have sequenced around 150 venom insulins. 40 of those are from fish hunters and look like the human hormone. And so lastly, I just want to show you one more thing that we did with one of these 40 insulins, and this was work done by Peter Ahorokome in my lab. And this is the insulin he studied, which is called coninsulin K1 from Conus kinoshitae. And you see that this insulin has this long A chain, which is something that had never been observed in any naturally occurring insulin or any insulin analog that had been described. And we knew from functional studies and from molecular simulations that this A chain was likely important for receptor activation. So based on this information, Danny made the next compound. So again, human insulin monomeric. And then he just put the A chain, this long A chain, on human insulin and a couple of additional mutations. And with that, we had created the next monomeric human insulin analog, again inspired by a different cone snail venom insulin that now we proposed uses a whole new molecular way of activating the human insulin receptor. And in a study published earlier this year, just a few months ago, we were able, by using studies on the entire ectodomain of the human insulin receptor, to really look at that elongation of the A chain and to show, and this is highlighted or circled here, and I point at it with my mouse here, that it is this long A chain that can come in and activate the human insulin receptor, where you would normally need that region of human insulin that also forms the dimer. So another cone snail had evolved another mechanism of receptor activation. And also in this study, we were able to capture the human insulin receptor in this asymmetric state, which had not been described before. And this was really beautifully shown, which we think is the active state of the receptor. And then this is the known symmetric state. And so, as I told you, there are more than 40 insulins from fish hunters. So I don't know how many more we will study, but clearly there would be more strategies of receptor activation to be discovered and more ways to make monomeric human insulin analogs. And if you want to read more about unusual insulins from cone snail venoms and also from other organisms such as viruses and also other venomous animals, we recently wrote a review on this topic that you may be interested in. And what I don't have time to talk to you about today is another doppelganger toxin, a whole family of doppelganger toxins called consomatins that we discovered since, that we are pursuing for the treatment of chronic pain and inflammation. And we have been able to gather some nice data on another doppelganger toxin. So with that, I want to thank everybody again who has been involved in this study, and thank you for your attention and for staying until the very last talk of today's session. Thank you.
Do we have any questions here in the audience? No, do we have any questions online? Oh, sorry, Glenn has a question.
Hey, Helena, Glenn King here. Fantastic talk. Fantastic as always. You've got two different ways of activating the receptor with different monomeric insulins now. Which do you think is going to be the better one from a therapeutic perspective?
Yeah, that's a very good question. And like I said, Glenn, it's not always the best compound, right? That makes it somewhere, as you probably know. So I think the second one is actually the better compound. And the reason for that is that the reason why the pharma industry also never really touched the monomeric insulin was that if you remove the B chain region, the compound becomes unstable. So we know that the first compound is actually fairly stable, but the second one is much more stable. It's much more stable than human insulin. And it's because it, at very, very high dose, at very high concentration, it forms a weak dimer. And so it's only at high concentration, which is fantastic for formulation. And then it very rapidly dissociates. But because the first compound has already advanced and has been licensed, the second one may not advance. So it's more, it's not a scientific reason why the first one might advance further than the second.
Thanks, Helena. Great talk.